Hi and welcome to my OCR A A-Level Biology revision session with me, Christine. So today's lesson I want to look at diabetes, which is part of your hormonal communication topic area. So to understand the impact that diabetes has, we first need to take a step back and remind ourselves about the role of the pancreas. So when we have high blood glucose concentration, the beta cells in the pancreas secrete insulin into the bloodstream, and that insulin will then make its way, and when it gets to a target cell, it will bind with receptors and a response will result. Now, those responses could be increased glucose absorption, it can be increased respiration, glycogenesis, lipogenesis. So what happens if someone has got diabetes? Well, there's two types. The first one we're going to talk about is type 1 diabetes. This is insulin dependent. So what this means is that the person has what's known as an autoimmune reaction. Their immune system is destroying their beta cells in their islets and Langerhans. And because of that, they're not able to produce their own insulin. And that becomes a real problem if their blood sugar levels get too high. So what can someone do if they have type 1 diabetes? Well, they can have the treatment where they inject themselves with insulin between 2 to 4 times a day. Now, the only way to know when to inject insulin is if they're actually monitoring their blood glucose concentration. Now, this can be done using different materials. Now, I've used a biosensor here. I pricked my finger, I had a tiny bit of blood that was then sensed through the biosensor, and it was able to give me a reading of 7.3 millimoles per litre. Now, our blood sugar levels should be controlled between 4 to 8 millimoles per litre, and therefore right now my blood sugar levels are within that range. So only if someone is getting to the higher range towards the 8 would they want to inject themselves with insulin. That insulin will obviously then be transported around the bloodstream, and that will result in the responses that are needed. However, it's a very fine balancing act because you need to ensure that the insulin that's injected doesn't result in hypoglycemia. If too much insulin is injected, then the blood sugar levels could drop too low. And if the blood sugar levels drop too low, that could result in someone becoming unconscious. There are certain parts in the body that require a constant supply of glucose because they're not able to store it, they're not able to hydrolyze fats, therefore they need to have glucose constantly being supplied for respiration. So it's really important that the blood sugar levels are maintained. They don't get too low, but also that they don't get too high. And that's where it's a very fine balancing act for someone who is type 1 diabetic. Now, with the advancement of technology and science, people now have machines that they can actually wear, which are continuously monitoring their blood glucose levels and administering the injections, the doses of insulin, when they are required. So what happens if someone's got type 2 diabetes then? So type 2 is called insulin independent. Now this is more to do with the fact that their glycoprotein receptors actually lose their responsiveness to insulin. It also links to an inadequate amount of insulin being produced by the beta cells, but it's more, if they're going to ask you a question, looking for you to understand that it's to do with the fact that these glycoproteins aren't shaped in the correct way, and therefore they're not able to bind with the insulin. So they've lost their responsiveness. So that links to proteins, a link to protein structure, and the fact that the glycoprotein is a carbohydrate attached to the protein. So they will try to interlink module 2 here if they want to go down the route of type 2 diabetes. Now, there is a lot of risk factors involved if you are type 2 diabetic. So people who are over the age of 40 and onwards tend to be more likely to end up with type 2 diabetes. So they're at a higher risk factor than someone who is young. Also, if you are obese, that could result in you having type 2 diabetes. Also, if you're eating a poor diet, if you have high blood pressure, or if you have a high blood cholesterol. So all of these things could result in this glycoprotein losing responsiveness to that insulin. So therefore, we need to think about, well, what treatment can somebody who has type 2 diabetes actually have? Well, that comes down to diet and exercise. So if somebody has type 2 diabetes, they're not going to be injecting because they're actually still producing the insulin. The problem is that their body's not responding. So they need to have low carbohydrate diets so that they are therefore not loading their blood plasma with lots of sugar. So they want to have low carbohydrate diets, or if they are having carbohydrate in their diet, that it is the polysaccharides that need to be hydrolyzed before they are able to be absorbed into the bloodstream. So a low carbohydrate diet, and obviously increasing the exercise. By increasing the exercise, there's going to be more respiration occurring, therefore the glucose will be utilized as the respiratory substrate. Now, for some people, the low carbohydrate diet and the regular exercise doesn't actually work. And if that's the case, they might need to actually take some drugs which will be used to either decrease the permeability of the glucose, or it could be looking at increasing the metabolic rate within the cell. So it really just depends if they're going to give you an example on what they would like as a treatment and why it might be necessary. So they love throwing out an evaluate question when it comes to type 1 and type 2 diabetes. Now, the key thing to remember with both diabetes is it can become a real problem if we have a high concentration of blood glucose concentration. That is called hyperglycemia. So if there's a high concentration of blood glucose, we have hyperglycemia. That is going to result in the movement of water from cells by osmosis. If the glucose is maintained within the blood vessels, water will leave the tissue fluid into the blood capillaries, and that will therefore cause a problem. Our cells will start to shrivel up, they won't be able to function properly, and also it results in this extra fluid in the blood which will then need to be filtered out by the kidneys. So someone who suffers from diabetes tends to urinate more frequently and produces large quantities of urine because of the blood sugar levels affecting their water potential, and that water needing to be excreted because it is in excess. Well, actually, it's not in excess. It's that it's being removed from the cells, and that becomes a real problem if someone is diabetic. So they need to make sure that they are constantly replenishing the water to try to maintain that water level. Also, if there's a lack of glucose in cells, that means that fats, then proteins, have to be metabolized. So remember, with your respiratory substrate, glucose is the respiratory substrate of choice. That's the best one. That's the one that's utilized through glycolysis. And if we need to utilize other molecules, then they have to be converted. They are energy demanding processes. So a lack of glucose in the cells means that fats, then proteins, but if we're breaking down our proteins, that results in organ damage. So that is a risk of both the diabetes. So it's important that the blood sugar levels are maintained within that narrow range. Remember, homeostasis is about maintaining this internal environment. So how exactly do we treat someone who has got type 1 diabetes and needs to inject themselves with insulin? Where does that insulin come from? Well, this is where your module 6 knowledge is going to come in the production of insulin from genetically modified bacteria. So I have done a whole video on genetic modification, but just to remind you, we can take a plasmid, a circular bit of DNA from a bacterium. We would isolate the gene, either isolate the gene itself from the DNA strand, or we can use mRNA, which would be treated with reverse transcriptase to make complementary DNA. What we can then do is we can use restriction enzymes called endonucleases, which will cut open both the plasmid at a very specific base sequence and the gene. And what that results in is these sticky ends where the complementary base pairs will come back together. Because remember, DNA is universal, the genetic code is universal, we can insert one gene to another organism by using DNA ligase. So we take the plasmid, we take the complementary DNA, and we can fuse them together using DNA ligase, forming those phosphodiester bonds. What do we then do? We take what's known as a recombinant plasmid. It's not the same as the original, so it's recombinant, and we want to introduce it into the host cells, and we introduce it by using what's known as a calcium-rich solution and increasing the temperature. So we do a bit of a heat shock to the bacterial cells. They will then take that recombinated plasmid into their cell, and then we grow that on antibiotic medium, and then it multiplies in a fermenter and produces insulin. And then we separate and we purify that human insulin. So it's a whole process that needs to occur, but it is very much linked to the fact that we can produce the insulin. So if someone is type 1 diabetic, they would inject themselves with the insulin that has been produced through the use of genetically modified bacteria. So that is linking this to your module 6 manipulating genome topic area. And the other treatment of diabetes is stem cells. So we know that stem cells have the capacity to go through mitotic division and differentiate. Well, there has been lots of research in the fact that stem cells can actually be reverted back to a potent stage previous to what they are now. So if they're multipotent, i.e. we're talking about our pancreatic cells, our beta cells, if we can use stem cells as a way of replacing those beta cells in the people with type 1 diabetes, that means that we could use this as a treatment. So this is a new research that has come out, and this is something that is moving forward. So if they're looking for you to understand how we use stem cell therapy for treating type 1 diabetes, it's about replacing those damaged beta cells by using stem cells, undifferentiated cells, which have the capacity to go through mitotic division and then differentiate. So it's really important that you do understand those two treatments that are potentially a way of helping someone who has type 1 diabetes. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, please do check out my revision platform, www.aiqchat.com.